Somewhere inside you, right now, something is rotting. Not in a way you can feel. Not in a way any doctor would flag on a routine checkup. But at the cellular level, in the trillions of tiny factories that keep you alive, waste is piling up. Broken proteins. Damaged machinery. Toxic sludge that your body was never designed to just let sit there. And here's the part that should genuinely unsettle you. Most people will go their entire lives without ever knowing there's a switch inside their own biology that can clear it all out. Not a pill. Not a procedure. Not something a pharmaceutical company patented and sells for $400 a month. Something your cells have known how to do for hundreds of millions of years, and something you've probably been accidentally switching off every single day of your adult life. By the end of this, you're going to understand exactly how that switch works, what triggers it, what shuts it down, and what actually happens inside your body when it runs at full power. And once you understand it, you'll never look at a bag of chips at 11 at night the same way again. Let's start with the problem, because you can't appreciate the solution until you understand how deep the problem actually goes. Every second of every day, your cells are working. Not resting, not idling. Working. They're manufacturing proteins, generating energy, responding to hormonal signals, repairing strands of damaged DNA. It's an unbelievable amount of activity happening in spaces so small you'd need an electron microscope to even see them. And like any machine that never stops running, things break. Proteins fold incorrectly and become useless, sometimes dangerous. Mitochondria, the tiny structures that generate the energy your cells run on, get damaged and start sputtering instead of producing clean power. Toxic byproducts build up in corners of the cell where they don't belong. Here's the problem. Unlike the trash you will out to the curb every Tuesday, this cellular debris doesn't just disappear on its own. It sits there. It accumulates. And over months, years, decades, that accumulation becomes something much more serious than clutter. Misfolded proteins start clumping together, interfering with normal operations the way a jammed printer brings an entire office to a halt. Damaged mitochondria begin leaking reactive oxygen species, unstable molecules that go on to damage even more of the cell around them, which triggers more damage, which triggers more leaking. It's a feedback loop, and it's one that researchers have tied directly to accelerated aging, chronic low-grade inflammation, and the early biological groundwork of some of the most feared diseases in modern medicine. So the question becomes obvious. If this damage is inevitable, and if it compounds over time, is there anything the body can actually do about it? It turns out there is. And it's more sophisticated than almost anyone realizes. Your body isn't defenseless against this decay. It has a system, not something engineered in a lab, not a modern biohack, but something that evolved over hundreds of millions of years and exists in nearly every cell you have. And for the overwhelming majority of human history, people activated this system constantly without even trying, for one simple reason: food wasn't sitting in a fridge 20 feet away at all hours of the day. Our ancestors went long stretches without eating not by choice, but by necessity. And in doing so, they unknowingly flipped this switch on, again and again, for their entire lives. The real question is whether you're still flipping it. Or whether modern life has quietly taken that switch away from you. In 2016, a Japanese cell biologist named Yoshinori Ohsumi received the Nobel Prize in Physiology or Medicine. Not for inventing a drug. Not for a surgical technique. He won it for describing, in painstaking molecular detail, a process he'd been chasing since the early 1990s, a process called autophagy. The word itself comes from Greek. Auto means self. Phagy means eating. Autophagy, quite literally, translates to the cell eating itself. That description sounds almost violent, maybe a little unsettling. But it doesn't come close to capturing how extraordinary this process actually is. What Ohsumi and the researchers who followed him discovered wasn't some crude form of cellular self-destruction. It's a highly organized, genetically regulated quality control system. The cell identifies which components are damaged or no longer needed, wraps them in a specialized double-layered membrane called an autophagosome, and ships them off to a structure called the lysosome. Think of it as the cell's recycling depot, where they're broken down and their raw materials get reused to build something new. This is not random destruction. It is precise. It is selective. The cell doesn't blindly consume itself. It targets exactly what needs to go, and it leaves everything else untouched. Before Osumi's work reshaped the field, scientists understood that autophagy existed, but they mostly viewed it as a last resort survival mechanism, something a starving cell did just to stay alive a little longer. What his research revealed was something far bigger. Autophagy isn't just an emergency response. It's a continuous, everyday quality control process, one that matters even when food is plentiful and the cell isn't under any obvious stress. When it's functioning properly, it sits at the very center of how your body prevents the kind of slow cellular decay that drives both aging and disease. That Nobel Prize wasn't a nod to an interesting curiosity buried in a lab somewhere. It was recognition that this mechanism might be one of the most important processes keeping you alive and functional, day after day, without you ever noticing it working. So how does your body know when to turn it on? Your body runs on signals, and one of the loudest, most influential signals it responds to is the presence, or absence, of nutrients, particularly glucose and amino acids. Every time you eat, especially carbohydrates and protein, your body releases insulin and activates a molecular pathway called mTOR, short for mechanistic target of rapamycin. Think of mTOR as a growth switch. When it's flipped on, it tells your cells to build, expand, and store energy. That's exactly what you want after a meal. But here's the catch: one of the very first things mTOR does when it's active is suppress autophagy. Growth and cleanup are, biologically speaking, opposites. You can't renovate a house while people are still actively building new rooms in it. This is where fasting enters the picture. When you stop eating, insulin levels drop, mTOR activity fades, and as those growth signals go quiet, a completely different pathway starts to rise. AMPK, which functions like a cellular energy sensor, constantly checking your fuel gauge. When AMPK detects that resources are getting low, it sounds an alarm that essentially says, "We need to be efficient now." And one of its primary responses to that alarm is to activate autophagy. The cell shifts out of growth mode and into maintenance mode. Instead of building new structures, it starts cleaning up the old, broken ones sitting around from years of wear and tear. This isn't speculation or wellness influencer theory. It's been documented repeatedly in human and animal studies published in some of the most rigorously peer-reviewed journals in the world, including Nature, Cell, and the New England Journal of Medicine. And here's what makes it particularly interesting: this switch doesn't require extreme, dramatic starvation to start moving. Your body is sensitive enough to the absence of food that even a moderate fast can begin nudging the balance toward cleanup. How far that balance shifts, though, depends heavily on how long the fast continues, your current metabolic state, and a handful of other variables. Which brings us directly to the question almost everyone asks the moment they learn any of this. How long do you actually have to fast before autophagy kicks in? The honest answer is more nuanced than what most quick answers online will tell you, and if you've heard a single specific number thrown around with total confidence, be skeptical of it. There is no magic hour. No switch that flips at exactly 16 hours and 0 minutes. What the actual research shows is a gradual curve, not an on-off toggle. In human studies, measurable increases in autophagy markers have shown up as early as 12 to 16 hours into a fast. But measurable and significant are two very different things, and this distinction matters enormously. Research using muscle biopsies and blood markers, including studies published in the journal Cell Metabolism, suggests that autophagy activity increases substantially somewhere between 24 and 48 hours of fasting in humans. Animal studies, which allow for far more invasive and precise measurement than anything ethical in humans, consistently show peak autophagy activity somewhere in the 24 to 72 hour range. One particularly striking piece of evidence comes from a 2019 study out of the Salk Institute, which found that a 24-hour fast produced dramatic increases in autophagy specifically within intestinal stem cells. Cells that are absolutely critical for gut health and tissue regeneration. And generally speaking, within physiological limits, the longer the fast continues, the more robust the activation tends to become. But there's a wrinkle here that complicates the simple headline. Individual variation matters enormously. Someone who exercises regularly, sleeps well, and maintains a lower baseline insulin level is likely to activate autophagy faster than someone dealing with insulin resistance or broader metabolic dysfunction. Age plays a role too. Autophagy efficiency naturally declines as we get older, and many researchers believe this decline is one of the underlying drivers of the aging process itself, not just a side effect of it. So while 16 hours is a reasonable, achievable starting point for most people, the deeper cellular cleaning that the research describes most compellingly tends to unfold further into a longer, more prolonged fast. Now let's actually zoom in and look at what this looks like at the microscopic level, because once you see it, you won't forget it. Picture a single one of your cells. You have roughly 37 trillion of them working together right now to keep you alive. Inside one of these cells, a membrane begins to form around a cluster of damaged proteins that have been sitting there, slowly gumming up the works. That membrane curves inward, wraps around the debris, and seals itself shut, forming a double-walled vesicle called an autophagosome. Under an electron microscope, it looks almost exactly like what it sounds like, a tiny bubble wrapping itself around cellular garbage. That autophagosome then travels through the interior of the cell until it reaches a lysosome, a compartment packed with powerful digestive enzymes. When the two structures fuse together, the enzymes get to work, breaking the contents down into their most basic building blocks, amino acids, fatty acids, nucleotides. Those raw materials don't just vanish. They're released back into the cell to be reused, recycled into whatever the cell needs to build next. This entire sequence is happening simultaneously, in multiple locations, inside a single cell, and it's happening across trillions of cells throughout your entire body at once. During a prolonged fast, the rate at which these autophagosomes form increases dramatically. Researchers have documented that in a fasted state, cells can process significantly more debris per unit of time compared to when they're in a well-fed state. And this isn't just tidying up for the sake of tidiness. The amino acids recovered from these degraded, broken down proteins get redirected to maintain critical functions, to synthesize brand new proteins, and to support the tissues your body prioritizes most heavily during periods of food scarcity, namely, the brain and the heart. There's one specific subtype of this process worth knowing by name, mitophagy. This is the selective removal of damaged mitochondria specifically, and it matters enormously because mitochondrial dysfunction shows up as a contributing factor in nearly every major age-related disease researchers study. When mitophagy is functioning efficiently, dysfunctional mitochondria get cleared out before they have the chance to leak reactive oxygen species and trigger those inflammatory cascades we talked about earlier. Healthy mitochondria then reproduce to take their place. The net result is a cell running on a cleaner, more efficient energy production system than it had before. This is a huge part of why researchers focused on longevity have become so fixated on autophagy in general. It's not simply about hauling out the garbage. It's about the cell rebuilding itself with something measurably better in its place. Now, what exactly does autophagy target? Because the list is more specific, and more medically significant, than most people ever realize. One of its primary targets is misfolded proteins, meaning proteins that fail to fold into their correct three-dimensional shape during or shortly after they were built. These aren't just non-functional leftovers sitting around uselessly. They're actively dangerous. Misfolded proteins tend to clump together, forming toxic aggregates that physically disrupt normal cellular operations. These aggregates are, in fact, a defining feature of Alzheimer's disease. The amyloid plaques and tau tangles that build up in the brains of Alzheimer's patients are, in part, the direct result of a failure in this protein clearance system. Beyond misfolded proteins, autophagy also goes after damaged organelles more broadly, not just mitochondria, but a stressed endoplasmic reticulum, dysfunctional peroxisomes, and even intracellular invaders like bacteria that have managed to sneak inside the cell. This last function has its own name, xenophagy, and it's part of why autophagy is actually considered a component of your innate immune system. Your cell can recognize an invading bacterium and package it for destruction using the exact same machinery it uses to clear out its own internal debris. There's also a category called agrephagy, the specific clearance of protein aggregates that have already clumped together and formed. This one matters enormously for neurodegenerative disease research, because the brain has an extremely limited capacity to replace lost neurons, which makes clearance mechanisms like this one especially critical for long-term brain health. A 2017 study published in Nature Communications demonstrated that enhancing autophagy in mouse models of Parkinson's disease significantly reduced the accumulation of alpha-synuclein, the specific protein responsible for forming the toxic Lewy bodies that characterize that disease. The implication here is enormous. Autophagy isn't some passive background maintenance task. When it's functioning at a high level, it actively pushes back against some of the most feared diseases in modern medicine. Which brings us to cancer, one of the most actively studied, and genuinely most complicated, relationships in this entire field. Early research showed that certain autophagy genes, particularly one called BECLIN-1, appear to function as tumor suppressors in specific contexts. Mice engineered to have reduced BECLIN-1 expression develop tumors at significantly higher rates than control groups. The mechanism researchers propose is fairly intuitive once you understand everything we've already covered. When cells lose their ability to efficiently clear out damaged components, DNA errors start accumulating, cellular stress climbs, and the biological conditions associated with malignant transformation become more favorable. A 2011 review published in Nature Reviews Cancer identified autophagy deficiency as a recurring feature across multiple cancer types in the models researchers had studied. The Alzheimer's research tells an equally striking story. Work coming out of the Nixon lab at NYU, among the most cited research groups in this entire field, documented that autophagy activity is severely impaired in the neurons of Alzheimer's patients. Researchers observed autophagosomes accumulating inside neurons without ever completing the degradation process, meaning cellular material was piling up instead of being cleared away. Follow-up studies in laboratory models found that interventions specifically aimed at restoring proper autophagy function were associated with reduced amyloid accumulation and measurable changes in cognitive markers under controlled experimental conditions. These findings have pushed autophagy into the center of several active drug development programs currently working their way through clinical trials. And then there's aging itself, where the data from multiple independent research groups all points in a remarkably consistent direction. In one widely cited experiment, researchers genetically enhanced autophagy capacity in mice and observed an extension of healthy lifespan somewhere in the range of 10 to 15%. Without any other intervention whatsoever. Separate research conducted in C. elegans, a small roundworm used extensively in longevity science because of how easily its genes can be manipulated, found something even more telling. When autophagy genes were deliberately blocked, the lifespan extension normally produced by caloric restriction disappeared entirely. That finding carries a specific and important implication. The longevity benefits long associated with eating less may actually operate, at least in significant part, through autophagy as the underlying mechanism. Across multiple model organisms, the data doesn't just suggest that autophagy is correlated with healthy aging. It suggests autophagy might be a required component of it. So if this system is this powerful, why isn't everyone using it constantly? Because the single most effective way to suppress autophagy is something almost all of us do without thinking twice: eating frequently. Every time you consume food, particularly anything that spikes insulin or activates mTOR, you're sending your cells a direct signal to stop cleaning and start building instead. In the context of a single meal, that's completely appropriate and exactly what should happen. The problem is what modern eating patterns have done to that balance. 3 meals a day plus snacks scattered across 14 to 16 waking hours means mTOR stays chronically elevated and autophagy stays chronically suppressed, almost around the clock. The cellular cleaning system that took hundreds of millions of years to evolve barely gets a window to switch on. Certain nutrients are especially potent at suppressing it. Leucine, an amino acid found abundantly in dairy, meat, and protein supplements, is one of the strongest known direct activators of mTOR. That doesn't mean protein is bad, it means that consuming it, especially in large quantities, will reliably shut autophagy down, at least temporarily. Glucose produces a similar suppressive effect through insulin signaling. This is exactly why some researchers studying autophagy remain skeptical of popular modified fasting approaches that include protein shakes or meaningful caloric intake during the fasting window. The molecular signal sent by those nutrients may substantially blunt the autophagy response, even if total calorie intake is technically reduced. Sleep deprivation is another underappreciated factor working against you. A 2019 study published in Nature Communications found that sleep loss specifically impairs autophagy in the brain, a genuinely alarming finding, given how critical neuronal autophagy is for clearing out the metabolic waste products that accumulate during your waking hours. The glymphatic system, your brain's own dedicated waste clearance mechanism, operates primarily during deep sleep and works in close coordination with autophagy at the cellular level. Chronic stress, through consistently elevated cortisol, suppresses these same pathways as well. And there's a real irony sitting underneath all of this: the exact lifestyle habits most associated with poor overall health, bad sleep, chronic stress, constant grazing throughout the day, are the very same habits keeping your body's most powerful internal cleaning system permanently switched off. So what does the research actually recommend, if you wanted to switch it back on deliberately? The most evidence-backed approach remains a combination of time-restricted eating and periodic, longer fasts. For baseline stimulation, compressing your eating window down to 8 or 10 hours a day, which effectively creates a 14 to 16-hour fast every single night without much extra effort, appears sufficient to produce measurable effects over time. For deeper, more substantial activation, the research points toward longer fasts in the 24 to 72-hour range, conducted periodically, whether monthly or quarterly depending on individual health status, as what's most consistently associated with significant autophagy upregulation in human studies. Exercise deserves far more credit here than it usually gets. A landmark 2012 study published in Nature by Beth Levine's research group at UT Southwestern demonstrated that exercise induces autophagy across multiple tissues simultaneously: muscle, liver, and brain, all at once. The underlying mechanism involves both AMPK activation and the direct mechanical stress placed on muscle fibers during exercise, which triggers selective autophagy of damaged contractile proteins. Interestingly, combining exercise with a fasted state appears to produce an additive effect, activating autophagy through two independent biological pathways at the same time. This is part of why morning exercise, performed before breaking an overnight fast, has drawn so much attention among researchers focused on longevity. A handful of specific compounds have also shown promise in this space. Spermidine, found naturally in wheat germ, aged cheese, and mushrooms, has been shown across multiple studies to induce autophagy through a pathway that's independent of mTOR inhibition entirely. A 2018 study published in Cell Reports found that higher dietary spermidine intake was associated with reduced mortality and better cognitive function in older adults. Resveratrol, found in red grapes and berries, activates a protein called CYRT1, a deacetylase that promotes autophagy-related gene expression. And metformin, a decades-old diabetes medication now being studied extensively for its potential longevity effects, activates AMPK and has demonstrated autophagy-inducing effects across multiple tissue types. None of these are substitutes for fasting on their own, but the research suggests they may work synergistically alongside it. And then there's the part of this story that almost nobody talks about: what happens after. When a period of significant autophagy activation ends and you finally eat again, mTOR reactivates, but now it's operating inside a cellular environment that's been thoroughly cleaned out. The raw materials recovered during the autophagy process become available for building brand new, fully functional proteins. Mitochondrial biogenesis, the production of new mitochondria, ramps up. Cells that just spent hours clearing out their damaged components are, in a very literal sense, refreshed. Operating more efficiently than they were before the fast even began. This refeeding phase isn't just a return to baseline. It's something considerably more interesting. Research in stem cell biology has shown that prolonged fasting followed by refeeding actually triggers stem cell activation across multiple tissues. A 2014 study out of Walter Longo's lab at USC, published in Cell Stem Cell, found that cycles of prolonged fasting caused immune system stem cells to regenerate, effectively producing a partial renewal of the entire immune system. Similar regenerative effects have been observed in intestinal stem cells as well. The implication is significant: the fast refeed cycle isn't just a cleaning event. It's a genuine regeneration cycle, where old, damaged components get removed and new, fully functional ones take their place. So here's the picture the research ultimately paints. Periodic fasting, and the autophagy process it activates, functions as a kind of built-in biological reset button. Not a cure for anything. Not a guarantee against disease or aging. But a fundamental maintenance process that the human body evolved over hundreds of millions of years to perform. One that modern life, with its constant availability of food, has largely disrupted without most people ever realizing it. The science doesn't promise immortality, and anyone who tells you it does is overselling it. But what it does suggest, consistently, across independent research groups and multiple model organisms studied over decades, is that keeping this system active might be one of the single most impactful things a person can do for their long-term cellular health. And unlike almost everything else in modern medicine, this one doesn't cost $1. It doesn't require a prescription. It doesn't require insurance approval. It just requires knowing it exists, and choosing to actually use it.